In the last lecture, we covered how natural mRNA behaves inside cells. In this lecture, we will focus on mRNA drugs. So here, the molecule is engineered and synthesized in vitro. So that's the fundamental difference. Okay, so we know that natural mRNA is made inside the cells by RNA polymerase II. And we also know that as it is being transcribed, it is capped, spliced, processed, all those things, and immediately packaged with RNA binding proteins. Whereas the mRNA drugs, that's completely different process. It is made in vitro, and then we use T7 RNA polymerase for that. So this enzyme, RNA T7 RNA polymerase, is actually a bacteriophage enzyme. And then, here there are no transcription factors or splicing machinery, cellular quality control systems, nothing of that sort are involved. So the RNA, which comes out is basically a naked molecule. It lacks a native co-transcriptional modifications. It is not packaged, and it is actually much more visible to innate immune sensors when injected, unless we engineer around that, of course. So what we need to be clear is that the stability, translation, immunogenicity is no longer biology driven. It is now chemistry and process driven. Therapeutic mRNA is made through an enzymatic workflow. So you will start with a DNA template, which carries a T7 promoter, and as we discussed before, T7 polymerase is used, and then it transcribes RNA directly from the template. So we already discussed that there are no transcription factors, no cellular context, right? So everything that cells normally do during transcription now has to be added deliberately. We introduce a cap either during transcription or enzymatically afterwards. And we need to define poly A tail, and then of course we purify to remove things like double strand RNA, which is one of the biggest drivers of innate immune activation. So the key idea is this: because natural processing is absent, every property of therapeutic mRNA is determined by engineered chemistry and process choices. This is one of the most important ideas in therapeutic mRNA. So a T7 polymerase doesn't know what natural RNA is. It simply incorporates whatever nucleoside triphosphates we put in the reaction. We supply ATP, GTP, CTP, and we get AGC. But for uridine, we have a choice. We can supply standard UTP, or we can replace it entirely with the pseudouridine or N1-methyl pseudouridine. So whatever we put in the reaction is what ends up in the final mRNA. So what we need to understand here is the NTP mix we provide is not just for transcription. It defines the chemical identity of the RNA we make. So here is a subtle but critical idea in therapeutic mRNA. We are actually working across essentially three different alphabets. First, there is the input alphabet, the digital sequence, FASTA files. So you are working with plasmids, codon optimized designs. That we generally talk as a TGC. Second, there is the IVT chemistry. And then for this, the alphabets are a bit different. And then again, T7 polymerase, it sees whatever nucleoside triphosphates we provide, and then that is actually a GC and whatever uridine variant we supply. So just standard U or pseudouridine or N1 methyl pseudouridine. And finally, there is this drug substance itself, a physical RNA molecule that's made of AGC and whatever uridine chemistry we decided to put. So these alphabets are obviously related, but they are not the same thing. So most modeling and design happens in the first alphabet, while actual biology and immunogenicity are determined by the third, that is drug substance. So the gap is where many failures come from. So you need to clearly understand these three alphabets. Um, so this is probably the single most important chemical intervention in therapeutic mRNA design. Replacing uridine with pseudouridine or N1 methyl pseudouridine fundamentally changes how the RNA is perceived by the innate immune system. So these modified bases strongly reduce TLR7 and 8 activation, and they mitigate REG1 signaling by altering RNA structure and reducing double-stranded RNA formation. So the downstream effects, if we see, are probably twofold. Immune sensing is dampened and translation efficiency increases because stress response pathways like PKR are no longer triggered. The RNA doesn't become chemically immortal, obviously, but its functional lifetime in cells increases dramatically because it's no longer being actively attacked. So for this reason, M1 methyl pseudouridine has become the default uridine chemistry for modern IVT mRNA therapeutics. Early therapeutic mRNA programs largely used unmodified uridine. So those molecules strongly activated innate immune sensors, which limited the protein expression and quickly ran into dose-limiting toxicity. And as a result, clinical uh, viability was tightly constrained. So modern mRNA platforms replaced uridine with N1 methyl pseudouridine. So that single chemical change suppresses immune sensing and improves translation efficiency and enables clinically viable dosing. This shift is a key reason platforms like Moderna and Pfizer's achieved a robust clinical efficacy. So the takeaway here is that the modified uridine is not a simple optimization step, but it is actually a necessity for therapeutic mRNA. Um, this slide actually explains why therapeutic mRNA needs so much engineering in the first place. Unlike cellular RNA polymerases, T7 polymerase operates in isolation because there is no processing machinery that normally converts a transcript into a mature mRNA. And then there is no natural capping, so a cap must be added. And then if that doesn't happen efficiently, the RNA retains 5' triphosphate end, which stimulates immune system. And there is no splicing, so coding sequence must be exon only. And there is no co-transcriptional polyadenylation, so the poly A tail has to be engineered in the plasmid sequence or can be added later on enzymatically. And because IVT reactions generate significant double-stranded RNA byproducts that never appear at these levels in cells. So, I mean, the key point is this. IVT mRNA is essentially chemically naked. If we want it to behave like regular biological mRNA, so every one of these missing features has to be engineered. Capping is one of the most critical process variables in IVT mRNA manufacturing. So there are two strategies. In co-transcriptional capping, a cap analog is included directly in the IVT reaction. When properly optimized, this approach produces a very high fraction of correctly oriented cap1 structures. Whereas in enzymatic capping, the cap is introduced after transcription. I mean, by using a specific enzyme. So this approach can achieve high cap1 levels. Its efficiency depends heavily on reaction control and enzyme performance. And conversion can leave mixtures of cap 0 or uncapped species. And why is this important? Because the innate immune system is sensitive to the 5' end. So if it's uncapped or improperly capped, RNA translation initiation gets blocked. So from a process standpoint, capping directly determines whether the mRNA will be translated or silenced. So now when it comes to poly we know that in cells, poly A tails are dynamically regulated and can range from roughly say 50 to some 200 nucleotides depending on the context, of course. So for therapeutic mRNA, so the style poly A tail has to be engineered. One approach is a template encoded poly A, where the tail is built directly into the plasmid. So this gives a deterministic length, but very long homopolymeric sequences can introduce plasmid instability during propagation. So the alternative is enzyme polyadenylation. So this is done after transcription. So I mean, this avoids plasmid issues, but produces a distribution of tail lengths. In practice, both strategies are used, and the choice reflects a trade-off between sequence control and process variability. Um, a, a major impurity that is unique to IVT is double-stranded RNA. T7 polymerase can generate double-strand RNA through several mechanisms you can see on the slide. So these double-stranded RNA are biologically active. They engage innate immune sensors, leading to cytokine production and suppression of translation eventually. So double-stranded RNA content is a defining IVT specific impurity and a critical variable that must be controlled through process design and downstream purification. All right, so let's see the summary of this lecture. Uh, Therapeutic IVT mRNA is fundamentally different from cellular mRNA. IVT mRNA is produced by a different enzyme, that is a T7 polymerase. And all this process relies on engineered chemistry, including those modified uridines we talked about. And then we also learned that 5' and 3' ends need to be engineered. And then IVT also produces impurity classes like double-stranded RNA. So because of all these things, even the primary sequence directly influences manufacturability, purity, and biological performance. So these differences between IVT and natural mRNA defines the engineering and modeling challenges we will focus on in the next lectures.